Hello, my name is Federico Lecumberry. I am an associate professor with the Signal Processing Department at the Engineering School, Universidad de la República. At the Signal Processing Department, we are 19 members working on several lines of research related with different kinds of signals such as audio, images, and video, machine learning or pattern recognition, computer vision, and information theory, applied to different domains and disciplines such as agro, biomedicine, and microscopy, among others. We have more than 20 years of experience in interdisciplinary research projects where we mix fundamental research and its applications. Luckily, we also have an extensive network of national and international collaborations. As a first glance of this kind of project we have developed, here I show you 5 examples of the combination of image processing and machine learning. The main idea from this slide is the power of the objective quantification and parameter extraction from images as an input to a machine learning system for classification, segmentation, prediction, or many other tasks. Related to bioimaging, we developed a system for subclinical disease prediction in milk cows based on ultrasound images, the detection of polyps in a 3-dimensional virtual colonoscopy reconstruction, or the melanoma detection in skin pigmented lesions from dermoscopic images. Also, the measurement of bovine beef quality parameters both in ultrasound and post-mortem RGB images. I will cover some of these projects later. However, the input information to a machine learning system could be computed from any kind of signal. For example, at left, the rhythmic pattern of playing a candombe drum is analyzed, detecting different styles associated with groups from different neighborhoods, or even the player style can be clustered and analyzed. Another general application of machine learning systems is the anomaly or outlier detection. In our group, we have worked with electrical energy consumption anomalies, trying to detect fraudulent scenarios, such as the one in red, in comparison with a normal time series. These methods could be applied to real-time analysis of communication links in mobile radio bases too. We have also developed outlier detection method for protein identification or viral evolution mutations based in genomic data. In microscopy images, we have been working with many collaborations. I had the chance to work in cryo-electron microscopy during my PhD, and I am still collaborating with them. Locally, fluorescence microscopy is one of the main techniques of application. We have developed methods for quantification in many different applications. In the picture, we can see an automatic system for cell parasite infection index computation, primary cilia length measurement in 3D, rodent somatosensory cortex quantification, and sperm tracking in fluorescence and bright field images. Also, we have worked with genomic sequence analysis, applying machine learning methods for viral evolution analysis and fusogenic protein identification. Before going further with some examples, let me give you a brief idea of what is machine learning and what we call deep learning. Let me use this definition of learning: improve the performance in a task with the experience. For example, playing some game, working with a machine, doing an experiment, or reading. When we apply this idea to computer algorithms, the task is the application we have in mind. For example, classification, prediction, segmentation. The improvement is a mathematical compute by some cost function, measuring a domain-specific similarity or some kind of error distance. And the experience comes with the data used by the algorithm. Trying to map this machine learning idea to the human way of learning, consider this child playing with these toys. When he grabs an object, he sends some properties. Let's say, measures some features, such as color, weight, shape, flavor, and with experience, learns that some pieces fit in one hole but not in the others. He recognizes some patterns in the object that helps to complete the task, and probably have a rewarding. Following this idea, the definition of machine learning, also known as pattern recognition, is automatically discover regularities in the data using computer algorithms, and from those regularities, take some action. Machine learning is a big field of fundamentals and methods. In the last 10 years, one particular class of algorithms are getting special attention and reaching the state of the art in many applications. These are the deep learning methods, which are based in artificial neural networks. An artificial neuron is a mathematical approximation or model to the natural neuron and its way of processing the electrical signals. The spectrum of machine learning methods is organized in these groups. First, we have the supervised methods, where each data point has a label, which is a class, positive or negative, for example, or a value, for example, the temperature or the daily count of positive COVID cases. When a new data point is analyzed by the model, a label is assigned. Typical applications are diagnosis, image classification, outlier detection, or forecasting. A second group of methods are the unsupervised ones, where no labels are known for the data points. In this case, the data is organized in groups or clusters given their similarities. Typical applications are high-dimensional data visualization or recommendation systems, such as the ones used by the online music or video platforms. In the middle of these two methods are the semi-supervised ones, where some data points have labels and some points not. And the last group of machine learning methods are known as reinforcement learning. Here we have an artificial software agent, a robot or a computer, taking a series of actions in order to solve a task. If the task is successfully achieved, a positive reward is received. If not, a negative reward is received. The typical application of these methods are in the area of game playing or robot navigation, and are more related to what we could identify as artificial intelligence, since the agent defines a protocol or policy to deal with a complex task. Two successes of this area are the AlphaStar or AlphaGo artificial players that recently beat the best human players of StarCraft and Go. In a traditional machine learning block diagram, we have certain steps defining this approach. Let's assume we are working with images. First, we have the pre-processing step, where an image is adapted or prepared, for example, performing some denoising or normalization. Then, we compute a group of characteristics or features describing different aspects of the data relevant to the particular task. Many times, these features are described and defined talking with a human expert in the domain. For example, a dermatologist has the experience identifying malignant skin pigmented lesions based on its color, shape, and uniformity, among others. Therefore, a mathematical representation of these features are computed, for example, color, border detection, shade descriptor, etc. These features define a multidimensional vector representing each image. These vectors are the input to the model training step, where the best parameters of the machine learning algorithm are obtained with an optimization procedure minimizing a cost function. This learned model is used when an unknown data point arrives to the training system. The same pre-processing and feature vector computation procedures are run, and the classifier assigns the label based in the experience represented by the model. If we don't have the labels for the data, the model and the last step are unsupervised algorithms instead of supervised ones. Usually, the data is split in three sets. First, the training set that we use in the model training. Second, the validation set that we use in the model validation step. And third, the test dataset that we use to compute the performance of this model. The deep learning approach is conceptually very different. As I said, it is based in the artificial neuron model, which is shown here, and different input xi, which can be seen as an n-dimensional vector x, are linearly combined with n-parameter or weights omega. This linear combination yj is the input to a non-linear activation function psi. For example, a thresholding with a parameter theta, which means that aj, the output, is 0 if y is less than theta, or 1 otherwise. In the case of an image, the values of the input vector could be the color of a pixel, or the intensities values in a region of interest around the pixel, for example. This artificial neuron is a basic computation block in the deep learning approach. Several of these artificial neurons are grouped following different architectures. Different layers of this array of neurons are organized in an artificial neural network. The term deep in deep learning is related with a high number of hidden layers in these networks. Now, given a training dataset, an optimization procedure computes the best parameter set that minimizes a custom function. This step is similar as in the previous traditional pipeline. However, an important difference you can notice in this architecture is that there is no dedicated layer to compute features. This is why in this approach, there is no feature engineering. The internal representation of the input data is learned by the network. On the other hand, the amount of parameters to compute is much higher than in the traditional approach. This leads to another characteristic of these networks. Usually, a huge amount of data is needed for training this high number of parameters. Also, it requires a huge computational power, usually using GPUs instead of CPUs. On the other hand, there are many open and reliable libraries for working with these architectures. Also, trained networks can be downloaded and adapt to a particular problem. In particular, in medical imaging, there are many available networks such as UNet, VNet, NiftyNet, and many more. Yesterday, I looked for recent bioimaging works in deep learning. Here I show you 4 examples. Top left is a semi-automatic interactive segmentation tool called Nuclick. The user clicks some part of the object, and then the trained network segments the object. This training was based in thousands on supervised segmentations. Top right and bottom left are 2 works on automatic image segmentation in a more controlled environment, for example, in magnetic resonance, skin lesions, and colonoscopy images. Finally, bottom down is a network that looks for increasing the resolution of a fluorescence image. Bottom row is the magnification of a select area. At the left, the original image. At the right is a structured illumination microscopic image. And in the middle are 2 outputs with different cost functions. The improvement in resolution is clear. These are recent works. Most of them have code you can download and test. However, they are not software in production, such as other software applications. However, Fishy is starting to add these kind of methods. Now that I hope you have a shallow idea of what is machine learning, let me show you some examples of bioimaging projects we have developed in the department. Here, a semi-automatic method for melanoma detection in dermoscopic images was tested. It reproduced the dermatology diagnosis procedure, looking at some features that can be easily computed from the lesion once it is segmented. In the image processing step, a hair removal and lesion segmentation is performed. In the feature extraction step, 57 features are computed, such as shape descriptors, edge regularities, color and texture, among others. This was computed from only 655 images, where only 111 had malignant lesions. These are much more lower number of images than other approaches based on deep learning. The results are very good and could be described as a similar performance as an expert. Clearly, this approach does not replace the doctor. However, it could work as a first screening tool. This work tests a complete system for polyp candidate detection in virtual colonoscopy. The 3D volume of the colon is reconstructed from 2D tomographic images. Then, the volumetric region of interest are defined based on texture and geometric features. This feature vector is the input to a support vector machine, a classical classifier from the machine learning toolkit. In these images, the top row shows 3 images of a real colonoscopy with 3 kinds of polyps. In the next rows, similar regions candidate for polyps are shown in the 3D reconstruct volume. The following works stress in some of the low-level image processing methods in microscopic images. In this work, we propose a method to automatize and systematize an objective identification of parasite and nuclei, and compute an infection index. THP1 cells were infected with Trypanosoma cruzi. The proposed procedure is shown in this block diagram. Stacked images contain 2 fluorescent channels, with pixels intensities proportional to the presence of parasites in green and cells nuclei in blue. The procedure was completely developed as a plugin for Fishy. Following the block diagram, C projection of the maximum intensity reduced the input data to 1 image. Median filtering reduced noise while maintaining edge information. Regions of interest are defined by binary mask obtained by automatic pixel intensity clustering, followed by operations to fill holes and close edges. The obtained regions of interest are filtered using shape descriptors such as area, circularity, and roughness. These segmentations are shown in figures B and C. In figure D, the segmentation of nuclei are shown in cyan, and parasites are shown in yellow. Since no information related to the cell's membrane is provided, a Voronoi diagram splits the whole image into regions based on the distance to nuclei edges. This diagram is shown as green curves in figure E. Finally, each parasite is associated to a nucleus, and the nucleus is labeled as infected. This let us compute a ratio of infection index. This process is repeated for all the stacks, and the statistics are reported to the user. Increasing the number of processed stacks improves the statistics and reduces the effect of errors in parasite association where no cell membrane information is present. At bottom, we show several images automatically processed. In this work, we use histochemistry techniques to allow the visualization of the neural somatosensory representation of rodent whiskers in the brain cortex. The emergent pattern is characterized by a matrix of dark patches, known as barrels, organized in rows of 4 to 9 barrels each. Here, we show a semi-automatic image processing method for segmentation and quantification of topological representation of whiskers in the somatosensory cortex. Images, such as the one in figure A, are taken through a binocular microscope and are contrast enhanced based in a hysteromycolization. The initial segmentation, shown in figure C, uses an automatic threshold for the detection of regions of interest. At this point, the user needs to make a manual selection of region of interest in a specified structure. This is the only step where the user interaction is needed. These regions define the initial curves for a curve evolution that adjusts to the barrel's geometry given the final segmentation. These are shown in figure E. The centroids of these regions defines the center of the grid that connect the barrels according to the user selection. This grid allows the definition of how regions are related as well as the measurement of selected parameters. Automatic quantification might include different measures such as grid orientation changes, intensity profile variation measurements, area, perimeter, shape descriptors, among others. To finish my talk, I just want to show you the web page of the Imagineer group we have at the Universidad de la República. This is focused in the acquisition, processing, and analysis of biomedical images. We also have collaborations from different institutions in Uruguay, such as the Instituto Pasteur de Montevideo or the Instituto de Investigaciones Biológicas Clemente Estable. This was what I had planned to present to you, so thank you very much, and if this is my email, if you want to contact me, and I would please to answer your question as far as I can. So thank you very much.